Excellent. Excited to hear about the discoveries you guys have figured out over here on the whiteboard. But thank you for being here for the panel discussion today. And in general, we talked about functional MRI. And I started in the field with functional MRI about 10 years ago, I think, maybe a little longer than that, 15 years ago, with my PhD training. And there's a lot of skepticism in the field at that time. And what are we really able to detect? Is there meaning to it? Is it noise? Is it really activity? And it's just been amazing to see things start to improve and improve. And I don't know if there's probably still a lot of skepticism, but it's definitely we're getting more specific signals. We're seeing more patterns that I think have a neurophysiological basis, and interesting to see the advancement just in the past few years. So, and that's through work with the field together. A lot of exciting things done by the people who presented today. So thank you for being here. So we talked about task-based functional MRI, resting state, functional MRI, animal imaging, and brain spinal cord imaging. This is an opportunity to talk about any sort of aspect around that, whether that's going to be on the acquisition side, on the processing side, ideas for cool projects in the future. We're going to invite Kawin Setsumpop to the panel, who has MR physics acquisition background. I'll let you just give a quick introduction.
All right, thank you. So thanks for inviting me to this. I normally do brain, but I have friends who do a lot of spinal cord, so it's good to be involved. So I do a lot of MRI acquisition and reconstruction methods. I've developed a bunch of stuff for fMRI and diffusion particularly, and as well as some structural imaging scan.
So this is an opportunity to convince Kawin to come over to the spinal cord imaging. We're trying very hard, and forcing you to be here today. But thank you very much. So let's just start off with any open questions that you'd like to ask the panel, and or discussions between yourselves. And maybe I could start off. With functional imaging of the spinal cord, I think the biggest challenge is shimming, and that's been discussed. What opportunities are there to improve it? Are there hardware options that we could do to help improve the shimming? And what does everybody feel here about that? Panel, any ideas? Yes, sir.
Okay, I'll start. I think there's some stuff related to like local shimming with the local coil array and stuff like that, and dynamic shimming. So I presume, and you can tell me, in terms of spinal cord imaging, you're having a lot of field variation, and that affects things in both ways in terms of distortion, but also through slice defacing and signal loss. So doing the shim part will help. Thinking about how to do encoding within the slice, making it thinner, but also try to encode within that slice so you have the thin slice resolution. Some of the stuff going back to what Gary did before on Hadamard encoding, maybe doing across the sub-slice there, so that you're getting rid of intravoxel dephasing, but still resolving those with good SNR. Those are some ideas, and then reduce field of view acquisition with EPI, try to reduce the field of view, more exotic RF excite with some of these fat saturation and better shimming component in both. Some stuff.
Yeah, sounds good. Sorry, I just want to make sure Alexandra is up and is also on the panel as well, and we're going to unmute Alexandra so she can contribute as well to the panel discussion.
Hi.
Are you contributing?
Yeah, I can say something. So I'm actually not quite sure if this is just the setup in our institute or whether this is a problem, but I think definitely the shimming still takes too long. So especially with our very long experiments, if you add at least half an hour to it, so that's always like around 2 hours for participants that they need to lie in the scanner. And I feel that often like the connection or communication between the PC that's running the scanner and the shimming procedures and things that need to be run basically, that could need some improvement. But as far as I understand, a lot of the things for security reasons cannot be run on the PC that controls the scanner. So we need to transfer data, run the shimming algorithm and so on. So I think, yeah, that's one issue.
Yeah, I was going to say that, you know, RF acquisition requires multi-coil stuff. So you need to get both the brain and the body, this part of the body at least, which requires some pretty complex RF coils. And there's a shove sort of recently that if you've got all these separate coils, which are wonderful for B1 shimming and B1 recovery of signal loss, you might as well use those same coils for B0 shimming as well. And so a number of people are looking at that, using these multi-coil things like Jones 56 channel coil. Why not use at least some of those coils also for B0 shimming? So make them also capable
Recording in progress.
of taking some DC currents and use them for the same reason of getting better B1, that is the RF's homogeneity. Why not also get better B0? And so that, I think, is going to prove really useful in the future. As I said, a number of people, including people here, have been looking at that. So there are opportunities for better hardware as well as better algorithms for reducing the B0, because as Christine and others have shown, you know, you have several hundred hertz of off resonance at 3T. We want to go to 7T, and then you have 7/3 times 200 hertz. So you really have to work at it for the higher field, which we're excited to go to, especially with our new Terra X machine that has awesome gradients. So.
I have not much to add from my side, but I would say usability of the shimming techniques, that would be very important, because if you lose half an hour per subject, it's difficult.
Yeah, and I would say the other challenge is then any sort of movement. So you spend all this time getting them set up, doing slice-wise shimming, and then they cough. Or I gotta adjust, you know, which is completely reasonable, but does cause challenges as well.
What's worse is I have to go to the bathroom and start over.
Yeah. Julian, any of the work you're doing, trying to with the shimming toolbox, and can that you see that helping reduce some of the time constraints and barriers?
Yes, I can say a few words, and then obviously Alexandre should take over because it's really his work. But to address the difficulty or like the challenge of having this accessible through the scanner, and you know, like what Alexandra was saying about cumbersomeness of having an external laptop and so on, is now solved on some vendors. So Philips enables to integrate some of the software and having, you know, Alex has shown a shimming toolbox running on Philips console, and now Siemens, as you know, has like Fire, which used to be which was a WIP, and now OpenRecon that you have access to on the XA61 platform, enables to run Docker container with custom code. And so Alex will show tomorrow some examples of how shimming toolbox could be run directly on the Siemens console. And so yes, those multi-coil, also called AC/DC coils, are we have very like positive experience with that. It does require, you know, hardware like love to put them together, and so it's not easily disseminable as slice-wise streaming is, because it does require the specific hardware. So that's a big cause. Then I had a question also that we've been wondering, playing with those dynamic shimming is so the using the second or even third order on the 70s. That's so it's not a thing because they are not shielded and they could create eddy currents. But is it possible to use just a little bit of current on those second order shim coils? Is managing eddy currents, but having the advantage of just correcting for each size. Is this at least possible through the software and through Siemens ecosystem?
I don't think it's possible now, but I think they're looking into it because the amplifier doesn't switch those things at that rate yet. So it's more of a static shim that you have to set and just keep. You can't update it as slice. But there's a direction that I've kind of been bugging them, and we're looking at some stuff. It's not a big solution. Yeah.
And the even order shims are inherently going to have long time constants. So the higher order stuff can only dynamically be done with like Z3 or some of the odd order things. Even orders have very large coupling with both the coils as well as the structures around, which so the high order shimming is inherently limited in dynamics to the odd orders, which means third order is the first one you can do dynamically.
All right, let's open it up.
I guess this is a more general question. So, an undergrad, I was in Alan Jasanoff's lab, and he was developing kind of contrast agents to detect basically other signals like serotonin or dopamine. And given that we know descending modulation is kind of acting to those systems, is there any thoughts about incorporating those type of contrast agents either in the preclinical models and other contexts to basically look at other signals in the spinal cord that might be relevant for processing?
Yeah, can anybody speak on other contrast agents?
Yeah, it's a really nice suggestion. We haven't actually tried out that, but we have thought on that. Yeah, the downward traveling signal and all are something that we are looking into right now. So I think your suggestion is something that we will take into in for future purposes. We haven't actually done any of those that you are suggesting right now. Yeah, but it's something that we will look into.
So I think, this was a point that was a bit brought up in some presentations this morning. So do you think that there could be a kind of generic protocol for EPI, for fMRI of the spinal cord? Or is this something that the difficulties with like shimming and also different like region of the spinal cord makes it too difficult? Maybe some thoughts about this.
I mean, it would be great, of course, but then, I mean, the difficulty maybe as compared to an anatomical protocol is with the functional protocol, you might have other constraints given the research question you want to address. And maybe for some questions, you do want to have higher spatial resolution. From some others, you don't care too much. So, I mean, then maybe we can have a repertoire of standardized protocols, but it's it's complicated. Like we have a study now where we do TMS on the brain and then simultaneous, or let's say interleaved cervical spinal cord fMRI. And again, we had other constraints given the TMS interference. So a generic protocol will be hard, but at least if something that we could use as a starting point would always be nice.
I guess just trying to come up with a agreement with spatial resolution is not so simple. The trade-off between resolution and TSNR and the length of the scan is hard. I think perhaps for motor experiment you may be able to do longer, but for pain is tricky.
Yeah, I also agree with both of them, and I think especially for animal, it becomes even more difficult because you are dealing with not only the small structure but a different species altogether. So the volume of FOV, everything will change, and also the questions that you are asking about is going to decide the acquisition protocol. But I think that common point of agreement around the basic sequences that we, for example, I mean, there are things related to global signal regression, and it is still like a 10-year-old debate that is still continuing in the brain community, and there have no consensus have been achieved till now. So I think like people should be allowed to use both the protocols and report their studies in a way so that people, the readers, can actually make their mind on that based on that. Yeah.
Oh, can I comment still? Okay.
My comment on that is that it's nice that people are sharing data sets. And so there's, just recently, there's another brain spinal cord data set made available. Not sure exactly, sorry, I forget what group made it available. Yeah.
China and Oxford.
Yeah, so there's another large data set available as we start to make more and more data sets available. We don't have, a comprehensive, sort of pre-processing steps. This does this, this affects this, with either resting state or task-based analyses, and that's something the field definitely needs.
I also had a comment on Sandrine's comment, so very good point. I feel like, and I might be playing a bit of devil's advocate, but the reason that people may not want to share functional protocols is also that because it takes a lot of time to probably prepare them, and they want to finish their experiments first before making it publicly available. I feel like, is that, what do you think? Is that a true observation? I mean, I feel like we can come up with a consensus, but maybe, like, if one group comes up with something themselves, would they really want to open it up? Is the other question.
Yeah. Any comments on that? Yeah, I think sometimes when they're more custom written sequences and not all product, it's I guess it depends. It's a lot of investment. I don't know. Thoughts on that and how to be more open with sharing protocols?
Yeah, I can direct my
Need
question.
to talk to my boss.
Maybe I can direct it to Julian to although it's today's panel, but yeah, what is your thought on that? Because you are leading a lot of open efforts. So what is your observation on that? Little bit of a controversial topic, but
So I guess it's a biased observation because we only observe the open protocols that are being shared because we don't see the ones that are not being shared. So maybe there is people developing stuff under the hood that we are not aware of, and we don't know it exists. My feeling is that the community, we have a good feel for the community, and I think that they are pretty open. And the major groups like Joe Kong, for example, opened that up recently, as you mentioned. I don't think people are necessarily very protective of their protocol. It's not like being aware of a protocol, you would basically set up like an fMRI experiment within 1 week, and then submit your manuscript. It's an extremely long process. That's my view. But who knows?
I have a observation, and that is that in the last few years, I have seen increasingly several journals have made it quite sometimes mandatory to actually share their data to the databases, and that actually or even for NIH different projects also they have made this that we have to share the data for our publications, and maybe accessibility to those shared data will help others to try out the original protocol and then their own protocol, and then if they see discrepancies, that can be one way of taking it up to the research community. Yeah.
Question is aimed more at Alexandra. I didn't have time this morning to ask the question, but I was wondering in all your pain experiments if you notice some kind of habituation or sensitization like in the spinal cord, and maybe is there ways in your protocol to kind of avoid these or to analyze these and detect them?
Well, this is actually a very good question. So behaviorally, that differs actually between participants. So some sensitize, some show habituations, but I think I never looked at the spinal cord over time. So I could not tell you whether the spinal cord in general shows a decrease over time or mirrors behavior or whatever. So yeah, maybe that's also a power question. If we would get reasonable results looking at time constants. But yeah, sure. I mean, I could easily set up a model that tests that. But then again, I mean, we don't know where this signal would come from, right? I mean, if it's due to the imaging itself, or if the signal in the spinal cord decreases, or if it's based on behavior. So yeah.
Yeah, so with an electrical stimulation protocol, we definitely see habituation. I'm going to show it tomorrow, the results. But now I have a question about between subject variability. So one thing that has thrown me off all the time when looking at when using different tasks is that even though we did exactly the same task, we see a large variability on the subject level results. Right, and exactly. Maybe Anirban, a question to you that you have this high resolution data or high quality data set, right? The gradient echo sequence, and a very well controlled task. Do you also see the same variability under these circumstances?
I mean, and animal studies, we are inherently limited by the number of subjects. That is one major thing that makes it a little challenging sometimes. I mean, the highest number of subjects that we sometimes have gone to is around 20. So that gives you an idea about it. But even within that, we have the chance of, because animal studies have a lot of longitudinal studies also, so that prohibits us from using too many animal samples in our study. I would like to mention here a few things that we have come across, and also in human also we are increasingly seeing is that the conventional notion that activation of particular regions will activate or stimulation of particular regions will activate only a certain network and not the other network. For example, if we stimulate a sensory network, we will see only in the sensory horns or the dorsal horns the activation. That theory is being challenged with every new paper that I am seeing nowadays, because there has been a recent study which has shown that for a particular task there is a cortical wide activation which is being seen And even in our data, we are seeing that although there is a specificity to relate to the region that we are targeting, but we do see activations which are varying at varying levels in different parts of the region, which suggests that the variability is not only between subjects but also within the structure itself. So it's increasingly making us questions about the networks which we typically think of are there are much more complexer networks than what we actually know. Maybe there are much more different levels of neural transmission that is going on in between. But overall, when you increase the number of sample size, the number of runs, and maybe remove some of the noisy runs and things like that, it shows up in some way or other the robustness of the actual experimental paradigm. Yeah.
Yeah, maybe if I may comment on that. So this inter-subject variability is very intriguing, and maybe it's because the activity is modulated by certain factors we don't know, we don't understand. So I mean, motor cortex. Maybe there is this. You have these beta-written events, and they actually influence. It's of course a completely different time scale than fMRI, but maybe those type of factors influence the activity we see with fMRI and explains the variability or part of it. But I think we see the tip of the iceberg.
I can just add, I haven't shown, or maybe I just showed one of the data in the morning presentation that we had actually done the motor cortex injury in 3 monkeys with similar kind of level of, like the idea was to have the similar level of injury, but always there is a little bit of difference. But what we saw is that even with the same injury model with all age sex matched monkeys, the recovery process was like very different for each of those 3 monkeys, and that kind of goes into saying how much variability we can see. The idea is that whether fMRI or other imaging metrics can pick up those variability or not, and can they be used to predict the different recovery outcomes and things like that.
Yeah, and I kind of, the within subject variability is, kind of just this, we're really struggling for signal, and sometimes, that peak, the iceberg peaks at one location, sometimes it peaks at the other location. Trying to collect as much data as possible. That's always been my goal with motor tasks or with some thermal tasks. Recently, though, we also tried to do this with electrical stimulation, and we found that we think there's a lot of habituation with the signal. And Sandrine's leading that analysis, and she submitted it to ISMRM as an abstract. Not sure if you guys have talked, but yeah, we collected a lot of data and it didn't help us, which is interesting.
But of course, just one more comment is that I mean there is a thresholding involved, right, in the functional MRI analysis. Then whether you threshold it at 0.01 or so. So it's also difficult to measure the similarity, right, because it depends how you threshold the subject level results.
Yeah, I think it also like doing, more of an ROI based analysis is also, can be useful, but it's also, we don't really understand the variability in the anatomy that well either. And we can do our best we can, but it's still a lot's uncertain.
So yeah, I'd like to bring back a controversial topic about the the resting state fMRI, and what we're seeing. Is it true? Is it partly true? And how much of it is true? And just to put that the context, it's about the studies of correlating points that are spatially very close by, and where the correlation could actually originate from physiological fluctuations, cardiac breathing, Meyer waves, and so on. And so Rob's actually point, which I could not answer, I don't know if Rob is connected, but one of his arguments is that they did a lot of regression, and then other groups reproduced these results. But to me, the reproducibility is not a proof that the effect originates from BOLD, because if it's vascular and it's fairly reproducible across people, then the experiment would be reproducible by different groups. To me, it's not a proof that this is pure BOLD. So I guess, I'm asking it again, can we think as a community of ways to increase our confidence that what we're seeing with resting state fMRI is truly BOLD signal? Can we think of, for example, the community of calibrated fMRI, where, they do a hypercapnic manipulation to adjust blood level, BOLD levels, for example. Are there ways where, we could be more convinced that this is true?
Maybe one take on this, because we can massage the data as much as we want. It's of course, if we can have some influence on how the signal is generated itself, that could help, as you suggest. But I think the brain plus spine is really very interesting, because, for instance, if we can infer spinal organization from the way it functionally connects with the brain, it's very unlikely that's explained by the same confounds, right?
Right.
So I'm really excited on that bigger field of view for that particular reason.
Yeah, I was more thinking of the bilateral blobs, for example, right and left, where if the vasculature is I would expect a lot of cross correlation in a small spatial location. That's seen also in animal studies, and yeah, I don't know how we can convince ourselves that.
Maybe your ending slides in your talk kind of got it, combining techniques. Like, is there a way we can combine, the functional MRI with other things?
I mean, so that has been shown that, because the brain resting state people have been attacked on that point, and they did some electrophysiology to show that those networks exist, and so. But we're talking at a different scale here.
I don't know whether this will answer your question or not, and I think it won't. But the hypercapnia challenge is something that is perfectly possible to do, and I also totally agree that there can be focused studies just to see whether the activations that we are seeing are actually from BOLD signal, or there are others, because otherwise all these efforts that we are putting in is maybe not actually meaning anything, and after 25 years of fMRI or 30 years of fMRI, we may come to the conclusion that resting state fMRI means nothing. But coming up with the actual alternative to actually study and remove all the other effects is the challenge. I mean, the challenge is not to question that thing, but also to think that whether what can be an alternative strategy to actually look into the BOLD fMRI signal without the effects of all the other motion and everything related artifacts. We have actually done one study and coming back to the question of white matter again, to see whether anesthesia actually affects the BOLD signal. And I forgot to mention this. I think I slightly mentioned this. What we saw is that the change in the white matter, for example, because of the anesthesia increase dosage, is similar to what we see in the gray matter, but it is not true in the control region, kind of countering the fact that that white matter modulation of the neural activity is also happening. It's not acting like as if white matter has no neural signal originating. And I know it's a very controversial comment, but that's coming back to the question that we have to select control regions and compare with them how much the connectivity are changing in the region of interest. Because if we see the activity in the control region also, then we can counter that these are maybe because of the surrounding physiological artifacts and things like that, which has been shown that it's not true. And I don't have too much of other ways, I think, in ways how we can tackle this.
Julian, correct me if I'm wrong. Some of your cat functional MRI this is task activation, right? Electrical stimulation, maybe? Yeah, okay. You showed the whole maps, I believe. Maybe? Did you show even outside the cord? You didn't necessarily crop to cord, right? And it's always interesting, like how much, activation was outside of the spinal cord, right? And what happens if we analyze the whole image? Like, where would we see connectivity across the whole image? Because we just crop out in our analyses, right? We do a spinal cord extraction or a brain extraction. I don't know. Would that help potentially get at, is there vasculature driving it? We're not analyzing the CSF signal or cropping that out. How much of the connectivity is in the CSF? Right?
What's your opinion on using, for instance, Pinnacle API images or anything which?
I think it's a good idea
because that will help us to go to the microvasculature. I mean, that's one way of.
I think it's a good idea.
Yeah.
So may I just join the discussion?
Yes, please.
I just wanted to say that I agree with Dimitri that probably brain and spinal cord help us to better understand this resting state activity in the spinal cord, and also the meaningful results of this connectivity, like meaningful connectivity between the sensory motor cortex and the spinal cord, like somatotopic organization, or when we are looking into like the gradients and seeing that the spinal cord is not a blob, it's really something within working currently with the brain. But I think that's something that we should also take care is the way that we denoise the data. And probably more and more people now are using slice-wise correction. And I think this is a way that we should denoise the data to remove for each slices respiratory, for example, noise or etc. And maybe not looking into the connectivity only at the slice level, but between slicings, it's more meaningful in terms of removing the noise effect if you're doing this way. And when we are looking into, for example, ICAPS with NAWAL and with medium granularity, what we found is not really left and right strong connectivity. It was true for C1, C2, C3, but for the segment related to the upper limb, it was really located at the right side or at the left side, so ventral dorsal connectivity, which for us was really meaningful because these segments are really related to upper limb innervation. And so really we expect a strong lateralization of the connectivity in this level compared to C1, C2, and C3, for example. So I think that the result help us to confirm that there is something meaningful in the resting state results.
The other conceptual aspect is we can exploit the time dimension still better, I think, than we do, because functional connectivity is, of course, by construction, an average over 10 minutes typically or something like that. So with ICAPS, because we do clustering in time, we somehow look at reoccurring patterns, and so the fact that they separate means things do not always happen over time the same way. Which, okay, it could still be driven by a confound that is mimicking this, but chances are getting lower. And just as a conceptual step forward, I think we should exploit the dimensions of our data in a better way. So,
and it's what you said about the spin echo reminded me also that using multi-echo could actually be very insightful. Because we know the bold sensitivity peaks at around TE of 30, and if you have multi-echo data, then you can check whether you see similar correlations at TE as like 10 milliseconds, and if so, it would make us more suspicious that those correlations actually originate from physio versus bold.
Christine, do you want to just comment on some of the challenges you've had with the multi-echo ICA?
Well, multi-echo is at the beginning, what I want to see is, can we see bold and non-bold signal in the cord? But we we can just reasonably get 3 echoes, which is not enough. And I'm hoping EPTI can be a solution.
Cowan, yeah. Tell, what is EPTI?
Okay.
They're only at 15 minutes. So I'm
All right.
I'm kidding.
2 sentences.
Yeah.
EPTI is something that came out of the lab where it's a time resolve version of EPI. So instead of just trying to get one image, you're trying to time resolve and get image at each echo time across the whole readout. So you theoretically you should be able to get an image at say 6 milliseconds, 7, 8, 9, and so forth up to like 40 or 50 milliseconds. So it's like a hyper version of multi-echo. And it involves a bit more reconstruction and modeling. And I think Nan from my group has been playing with it with you guys. Hope to try to help you on some of that. Yeah. It's 2 minutes, see?
You still have 13 more minutes. I was really going to give you the whole 15. But yeah, looking into this as a possible benefits from it. There's also been with the multi-echo ICADOs, there's issues with it converging, right? And getting a solution.
Yeah, it's not so simple. Yeah.
And you've been working with the Tadana people trying to get feedback on how to improve it.
Slowly.
Yeah. So.
It's hard when you have 3 echoes. Putting an exponential with 3 echoes is not.
Much easier.
But the images look good. Quality at least, relative. Additional questions? Anything online?
So, yeah.
Oh, yeah.
Ani, I've been wondering, you mentioned that anesthesia reduces the bold signal, right? How does it affect the physiological, like the cardiac cycle, the signal coming from the cardiac and the breathing cycle? So could this actually help disentangle the contributions of the bold signal and the noise in the correlations?
Not to my knowledge, because I think that's a fair point that we have mentioned, and we actually monitor the cardiac respiratory patterns regularly during the entire period of the study, because that's part of the protocol. And the animals actually have a much higher heart rate and cardiac cycle compared to the human.
Yeah.
And we actually, what we know is that if we the cardiac levels goes very high, that means the anesthesia is actually too much. Too less. So we try to bring it to a range. I mean, they are proportional in that way, but it is much consistent or within a particular range of the isoflurane amount. So we don't observe too much fluctuation within a certain range of isoflurane, and all our studies are relative to that, as you can think of it. I mean, if you change the isoflurane level, we have seen going to a lesser isoflurane, the cardiac rate and everything will go up. So that's something we take into consideration. More importantly, the respiration, we are able to do it mechanically using a mechanical ventilator. So we can keep the respiration at a particular rate, which is also much higher than humans. We have 60 cycles approximately in animals. But that helps us because we can keep them. As I said, animal studies, we can do a lot of control in various aspects. So we can keep the respiration cycle same and things like that.
But did I understand you correctly? So the anesthesia reduces the correlations in the gray matter.
I think I haven't done the white matter, but the paper that I referred to has shown it in the gray matter. But again, that's in brain, not a spinal cord paper. But in brain, we have seen definitely that there is reduced correlation. That the pattern of the changes are consistent across regions. Yeah.
You haven't looked at the spinal cord, correct?
No. That's.
Would that be challenging study to do, or could you do it quite easily, like on top of another study? I don't do any animal
I
stuff.
don't think it should be challenging because we regularly modify our isoflurane level based on whether the animal is getting too deep or getting awake or not, because we can figure it out using the cardiac cycle and things like that. So I don't think that's just. I mean, we thought that okay, the brain study may be enough to make people convinced that anesthesia has some effect, but it is a consistent thing. I think that can be definitely one project that we can do just to make sure that it is also similar to the brain regions.
What about faster imaging? Like if we could go fast enough to capture cardiac noise and then be able to filter it out. So if we did a single slice, Julian, as fast as we could, zoomed it in, collected 6 minutes of data, we're able to filter it temporally, but still show that the left ventral horn's talking to the right ventral horn, or left dorsal horn's talking to the right dorsal horn. Would that be more convincing? I mean, there might be issues with it.
The question then is the filtering, because when we regress out things, and I was telling chatting. So I mean, I could use the whiteboard, but essentially, if there is imagine that the cardiac signal is periodic, but it's periodic, but it's also modulated by lower frequencies things. So the amplitude would change. If you regress this out at a particular frequency because you can afford it, you would still see some bumps up and down because the amplitude of the peaks is not the same.
Yeah.
So there is a regression, but you still end up with non-negligible variance that you would then correlate between voxels. So you could mitigate the problem. I don't think you can get rid of the problem.
The question is an order of magnitude thing. How much are you regressing out? If you're regressing out 95% of it, then it's probably going to help a lot. But if you're regressing out 2/3, then that's a problem. You still have a lot.
If you go very fast, then you can do fluids. And then that amplitude modulation on the fast frequency is in the width of that high frequency component. So if you go really, really fast and completely take it out, I think you should be.
So you would do like a bad pass band, but okay. And how about the harmonics and the fact that the cardiac?
So it's an order of how much left, right? And I think if you go fast enough, you can probably capture 95% of what you need to kill.
Can PTR help with the TR?
With the TR?
Well, yeah, faster image.
It's a bit difficult because you're trying to do with ePTI. What you're trying to do is you're trying to get more information, meaning that you're trying to get images that are not distorted as well as across multiple echo. And right now, what we're trying to do is we can get that, but of course, it's going to take longer than a normal acquisition, and we'll be speeding it up. And I would go so far as we're trying to get to the same time as the normal single-shot EPI acquisition to give you this extra information. I'm not sure I can go a bit faster than that still. If you want me to do the normal EPI, there are ways to try to speed things up, but I can't give you everything quite.
So I have a question. So we have here experts from both more on the acquisition side and more on the post-processing side. Which do you think is more important? Do you think it is? Do you think it is? For example, we can do the breathing correction. I don't know all the details, but sorry. Yeah, and the acquisition side, but also post-processing side, right? Like which one is? And I know they are challenging, but which one is more important?
I think it's important that they talk. Because then you kind of know the problem a little bit more. And for me, when I develop stuff over the years, it's both the hardware, the acquisition, the reconstruction, and the processing, as well as the application of it. And if you know all those steps and what are the bottleneck, it kind of helps a bit more to work through more. So it's more important that they talk.
I agree. But also, if you don't have good data to begin with,
you can start at the front.
As far as I know, we're not employing any. Pretty certain actually. We're not employing any deep learning reconstruction with our functional imaging. And is that possible with the Zoomit gradient EPI sequence? And is there a reason not to do that? Or I don't know.
The manufacturers are very conservative about that because of all this discussion about hallucination and so on. So the deep learning stuff that you can get on the manufacturers' machines is based mostly on denoising as opposed to finding more subtle stuff, increasing the resolution or whatever. So it's pretty conservative in that sense that it's mostly denoising, and that's just based on a fairly conservative approach to reconstruction of not trying to get very, very high resolution out of a crummy acquisition of 3 millimeter isotropic or something and making a 1 millimeter picture out of that. So I don't see a lot of that kind of reconstruction improvement coming along, even with generative AI and all of the stuff that entails.
Thanks, Gary. Any additional questions? I've got one. So, Julie, maybe this kind of is tailored to you, but I know you mentioned we talked about trying to do isotropic functional imaging for the spinal cord. We're usually like 1 by 1 by 3, 1.25 by 1.25 by 4. There makes a good point to try to go towards isotropic. I know you guys tried it, and kind of just curious, it didn't work very well, right? Or did it?
Actually, it did.
Oh, it did? Wait, it worked well? Or what?
Yeah, so we're trying to explore that avenue.
Okay.
And see whether it actually makes things better, because it's a trade-off. It's true that acquiring with bigger voxels is more efficient in terms of getting more SNR as opposed to averaging later on. On the other hand, you also get less intravoxel dephasing, and you could gain more by getting with smaller voxels from that respect. But it has to do also with the quality of shimming. So these are the concepts that need to talk with each other, and then based on the shimming, it's the strategy of question of what is the voxel size that is the most optimal. But I think there's interesting avenues there.
Okay. Well, yeah. What kind of voxel size you think we should do? Is there, if we're doing isotropic, do you want 2 by 2 by 2? Just curious. Any thoughts? Do you have any feelings?
Well, if it is 2 by 2 by 2, is standard brain voxel size.
Yeah. I'm just saying. I don't know.
Well, I'm curious about the isotropic. Is what you're saying? What, 1 by 1 by 1?
We've been trying at 70.
Okay.
Okay, 70. Because we're talking about TSNR. If you're doing 1 by 1 by 1, you get 10% of the TSNR. I'm curious how you see activation.
Yeah. The other thing is that if you reduce the volume by, say, a factor of 2, it takes you 4 times as long to scan that and get the same SNR. So, it's a pretty heavy. So it kind of depends on how additional z-axis resolution helps you. If everything is uniform within 1 or 2 or 3 millimeters, then 1 or 2 or 3 millimeters is probably the optimum size to acquire.
Uniform in terms of volts in them, or in
terms of function? In terms of function, I mean, if there's no difference between 1 millimeter and 1 millimeter next to it, then you might as well acquire 2 millimeters instead of 2 1 millimeters, because it'll take you less time to get the same SNR. So I mean, there are huge trade-offs, and as you know well.
But that's assuming that there is no intravoxel dephasing.
Yeah, well.
Great. Cool. Okay. All right. Any other questions? Oh, great.
This is again slightly focused on pain. I guess I've learned a little bit about MR spectroscopy, and I was really interested in whether I guess all the pain people have tested out the metabolite profile, and also while causing pain, I guess, in the brain or the spinal cord. I think there's some work on glutamate NAA values and how they relate to general recovery from nerve injuries. I don't know if there's any work that you guys have performed. Okay, cool. Thank you.
I cannot answer the specifics about it, but our group has tried on animal models and looked into different other imaging sequences. Like they have looked into CEST imaging and QMT and others in the spinal cord, and found certain metabolites and things which may be quite specific to the injury-related changes. Also, the myelin content and things like that. So yeah, there are groups also who have done this apart from us, but in our group, I think in rat models of injury, we have done this. Yeah.
Mm-hmm. All right. Oh, go ahead.
Yeah, I don't know precisely at the spinal cord level and spectroscopy. I would need to look it up. But at the level of the brain, the spectroscopic imaging is getting a revival now. So there's a lot of work on having an image of these different metabolites with like eccentric and rosette and this type of sequences. So maybe some of this might be brought down to the level of the spine. It would be very exciting.
I think it's always a good go to OHBM, see what everybody's doing, and then try to do it in the spinal cord. That's it's a good strategy. And then do it in the cervical spinal cord, and then try to do it in the lumbosacral cord. I think that's it's a really good strategy. Yeah, and then back up, turn around. Any additional questions for the panel? If not, go ahead.
Question? So actually, we have done some spectroscopy in the spinal cord in our lab. I'm not so sure about the current status of the project. So this was in healthy subjects, no injuries, but I think it was way more difficult than thought. And the experiments took a very, very long, I think 2 hours. And that as you have to define one voxel that is scanned the entire 2 hours, people are not allowed to move, or they should, of course, not move. And you can imagine for 2 hours lying in the scanner, this poses some challenges. So I'm not sure what the current status is, if there will be a publication, but yeah, difficult.
Yeah, I know a group about 15 years ago, they were piloting in whiplash injury, post-injury, and they've never published anything from it. So it was probably quite challenging.
But it's spectroscopic.
Oh, yeah.
Yeah, I can just confirm that it's challenging. We did one study in spinal cord injury patients at level C2, C3, above the level of injury, and healthy controls, just to see how the metabolic profile changes after injury. Because, there's some neurodegenerative changes, and we saw some markers of degeneration and neuroinflammation. But the interpretation is a bit more challenging because you have to standardize according to other metabolites and cannot really have absolute measures. So those were relative measures with choline over myoinositol, for example. Just interpretation gets a bit more complicated because you cannot have absolute measures there. But also in terms of getting the data, it was long scans. But there's not much, not much in the spinal cord. One very recent paper also in spinal cord injury from the Zurich group in the lumbar cord and M1, where they looked at the metabolic changes, but there's not much more.
All right. Any additional questions, comments? Okay. All right. Well, thank you so much for being here, panelists. Thanks for everybody's great questions. Today was good. I think we maxed out the time for every single talk with questions, so that was better than yesterday. Let's do the same tomorrow, and we're going to start next in here, Merve, with the hands-on session. Is there a little break, or are we going to go right into it?
We can take a little break. It's a long session.
It's a long session. So let's do a 10-minute break. We'll start at 3:10.